Okay, so now we're going to take that molecule pyruvate that we created earlier via the glycolytic pathway. Remember, this is 2 times 3 carbons here. And then we're going to break it down. So we're going to completely generate 6 molecules of carbon dioxide there. So all the carbon that came from that glucose will be broken down by the end of this process. And we'll end up pulling out from the potential energy representing that pyruvate multiple molecules of NADH, FADH2, which are reduced electron carriers. And remember, there's bucket loads of energy there, and we'll make a teeny bit of ATP via GTP in this process. So the result, when we're done with this, we'll have created all of the, we'll have completely broken down the original sugar in the carbon dioxide. This is where the carbon dioxide comes that we breathe out. And then we will have taken the electrons from that, shoved them into NADH2 and NADH and FADH2, and made a little bit of ATP out of this process. So here's where we are. We've captured just a little bit of this energy thus far. The process of going from glucose to pyruvate, negative 146 kilojoules per mole. But if we can capture all of that, then we can get, what is that, 20 times more energy that is produced there. And so here we are. We went through this glycolytic process in the last chapter, and then we're going to look at this citrate cycle next, and then we'll do oxidative phosphorylation at the end. Oh yeah, Krebs cycle, the TCA cycle, the citric acid cycle, all kind of refer to the same thing. And remember, from our Gibbs free energy in glycolysis, we took a molecule of glucose that had, and we kind of normalized it here, but it has a potential energy associated with it. We activated it with 2 molecules of ATP, split it, and then withdrew the energy here to form 4 molecules of ATP and NADH, and then we have this pyruvate. This pyruvate is of lower energy than the glucose, but there's still a lot of energy left, and we'll see that at the end. So in order to get to the citric acid cycle, one, oxygen needs to be present. So this is an aerobic mechanism, even though we don't technically use oxygen in the citric acid cycle. And this occurs in the matrix of the mitochondria. So glycolysis occurs in the cytoplasm. Here, this occurs in the interior of the mitochondria. Both pyruvate oxidation and the citric acid cycle occur there. And we will see there are links, direct links from the citric acid cycle to oxidative phosphorylation and electron transport chain that we'll see in time. Here, the function of pyruvate oxidation is to make these acetyl-CoA units. I said before that everything goes back to acetyl-CoA, and each of these acetyl-CoAs will generate 3 NADH, 1 FADH2, and 1 GTP that gets converted to ATP. And remember, you're going to generate 2 acetyl-CoAs from the 1 glucose molecule that was started with, and we're going to get a little bit more NADH from the pyruvate oxidation process. So pyruvate oxidation is really preparing for the citric acid cycle or the Krebs cycle. Here we take this 3-carbon, 1, 2, 3-carbon pyruvate. We are going to decarboxylate it, so we're going to remove a carbon dioxide from it. Every time in this process we remove a carbon dioxide, we're going to reduce a molecule of NAD plus to form NADH. So every time we generate we release a carbon dioxide, we make an NADH there. And we're now left with 2 carbons in the form of an acetyl group, and that is going to attach to a coenzyme A to form acetyl-CoA groups. Again, catabolism converges on acetyl-CoA, so this is one process to make acetyl-CoA. There's others, when we talk about beta oxidation fatty acids and things like that, that can also produce acetyl-CoAs. And this is the kind of, bio for non-majors view of the Krebs cycle. And again, you get to this, or at least a bio 101 view, then you can get pretty far. We have a 4-carbon oxaloacetate, 1, 2, 3, 4 carbons of oxaloacetate. We're going to add the 2 carbons from the acetyl group to form a 6-carbon molecule, citrate. And we're going to take that 6-carbon molecule and we're going to remove 2 carbons from it. So we'll do a round, remove a carbon dioxide to form a 5-carbon structure, alpha-ketoglutarate, and then we do it again to form succinate. Every time we remove a carbon dioxide, we generate a molecule of NADH. We're left with a 4-carbon molecule, and then we're going to oxidize that 4-carbon molecule back to oxaloacetate and pull out some energy there. So this is an 8-step process, enzymatic step process, but essentially that's all we're doing. Take a 4-carbon structure, add 2 carbons to it to form a 6-carbon structure, and undergo 2 rounds of decarboxylation to reform a 4-carbon structure, and then oxidize that molecule in order to form oxaloacetate. The reduced electron carriers that we get will go into the electron transport chain where we can get a massive payoff of energy from it. So the acetyl group from this coenzyme A initiates the process. This coenzyme A, it's a coenzyme, so it gets chopped off and it gets recycled, both later on in the citric acid cycle and then also in pyruvate oxidation. And then the cycle is in steady state, so we're adding atoms into the cycle, but the concentrations of the intermediates, citrate, the acid citrate, things like that, they don't change at all. So the result of this process, carbon dioxide, these reduced electron carriers, which again is where all the energy is, and a little bit of ATP. And this is a look of the citric acid cycle. Again, for biochemistry, we need to know the structure of the intermediates, the enzymes that catalyze the reaction, and what's going on. And again, just like with glycolysis and with the Michaelis-Menten equation, you don't have to memorize 8 different intermediates. If you know the structure of oxaloacetate and you know what's going on, then it's kind of trivial. Take oxaloacetate, we add 2 carbons to it. And you can see that 1, 2, 3, 4 carbons. 1, 2, 3, 4 carbons here. We're adding 2 carbons, so 1, 2, 3, 4, 5, 6 carbons there to form a 6-carbon molecule. And then we're removing carbons, 1, 2, 3, 4, 5 here as 5 carbons. And 1, 2, 3, 4 here. So we're removing a couple of carbons. Every time we do, we form an NADH, and then we oxidize this 4-carbon structure to get back to oxaloacetate. And so knowing the chemistry gets you a lot further, and you'll kind of understand this process a lot more than simply brute force memorizing 8 different structures. And if we look at the delta G, we have a glucose again. We activate that glucose to destabilize it. We split it, pull the energy out. So pyruvate, which we're starting with, is of lower energy than that glucose. But note there's still a lot of energy left on the table here. And so we're going to break that down to oxaloacetate here, much lower energy than this pyruvate. And so we're going to extract and capture as much as we can as NADH and as ATP. And this is wrong. Each glucose would yield 6 carbon dioxide, 10 NADH, FADH2, and ATP. And there's some water in there too, but I don't know what happened to that. Okay, so the first step is to prep pyruvate. to form acetyl-CoA. The enzyme complex that does that is pyruvate dehydrogenase. This is a 3 enzyme complex that are all together in a kind of factory change, and it requires a bunch of coenzymes. Remember, coenzymes are necessary for function, but can be changed themselves. So it requires NAD+, FAD, coenzyme A, thiamine pyrophosphate, and alpha lipoic acid. And so we'll use all of this in order to break down this pyruvate dehydrogenase. And remember, NAD and FAD, or electron carriers, they have the ability to pick up electrons and move them someplace else. And so those electrons will come in the form in these carriers' case, in the form of hydrogen atoms. So here is NAD, and then here is FAD. It does the same thing. FAD, so NAD picks up a pair, 2 electrons. FAD can pick up electrons individually to form a semiquinone or a pair of electrons over there. Coenzyme A is a modified nucleic acid, and essentially it has this long chain that ends with this thioester here. And this thioester is used to preserve the energy of the acetyl group. And so the acetyl group attaches here. So this is a 2-carbon group that attaches via this thioester bond over there. And so all of this coenzyme is basically a presentation pedestal for the acetyl group, and the energy of that is preserved via this thioester bond. TPP is another modified nucleic acid, and it has this pyrophosphate group on it, and it'll be used to move that acetyl group through the process. And then alpha lipoic acid is a lipolysine, and so you have this lipoic acid group that's covalently attached to a lysine group. So this would be an example of a prosthetic group. I can't spell that, but you know what I'm talking about. And it has the ability to be either oxidized, it can be acetylated, and so here this is a 2-carbon acetyl group, so you can imagine kind of what an acetylated dihydrolipoamide will do. It's moving the acetyl group from coenzyme A, and then I don't have an image here. It can also be fully reduced. And then that would simply replace that acetyl group with another electron pair there. And I think those are the 5 coenzymes. This is a look of the pyruvate oxidation complex. So note this is enzyme 1, 2, and 3. And we didn't really get into symmetry here, but you can see that there is a symmetry of this complex. So it goes here, here, and there. And then there's another group that comes in. And so this allows for the products of one enzyme to go directly into the next process, so that there is there's no side reactions that occur or loss of enzymatic functionality. And then this is a reaction. Simple, huh? Take this pyruvate. Note this pyruvate. Remember we said the carbon dioxides, they are waste products, right? So they're completely oxidized, and they don't have a lot of energy to them, right? It's poor spelling there. So we see this nice little carbon dioxide group on this pyruvate, right? We're just going to chop that off. Again, we're going to use coenzyme A, we're going to use NAD, FAD, TPP, lipolysine, and then we're going to generate an NADH. So whenever we remove a carbon dioxide, we're going to reduce NAD plus to form NADH. We're going to generate a carbon dioxide, and we're going to generate an acetylated coenzyme A. All right, and note this thioester is really important, so this is the acetyl group, and then it is linked to that coenzyme A. And the pyruvate dehydrogenase process is the 3 enzymes that are going to make that happen. And here's a look. So again, pyruvate, coenzyme A, NAD+, you generate acetyl-CoA, carbon dioxide, NADH, a little bit of energy as well. Note the coenzymes, so the 3 enzymes, E1, 2, and 3, they have to be regenerated in the process, but the coenzyme and NAD, coenzyme A and NAD+ can be changed in this process. So NADH is formed, acetyl-CoA is formed. They're obviously different from the substrates there. And this is the process, and it looks kind of complicated, but it really isn't. You can imagine this is kind of going through time. So maybe the next iteration of this all animated or something like that, or maybe they have some that animate it. So what I want us to do is just to travel with this pyruvate molecule here so that we see what's going on. And so we have our 3-carbon pyruvate molecule. It links up with TPP. In the process, we lose the carbon dioxide. So now we're down to 2 carbons, 1, 2, right? That represent the acetyl group here. It is going to link up with TPP, and TPP is basically going to carry it to this lipolysine. So the lipolysine is oxidized. It has these sulfur sulfahedral groups that are linked to each other, and then it is going to pick up the acetyl group to form an acyl lipolysine. So here's the acetyl group, right? This is an acetylated lipolysine, and it's simply going to swing around and carry that acetyl group to coenzyme A. So form the acetyl group, TPP picks it up, takes it to lipolysine, lipolysine picks it up, becomes acetylated, brings it to coenzyme A to form acetyl-CoA. So now we've generated our acetyl-CoA. When the coenzyme A picks up the acetyl group, it actually reduces the sulfahedral group. So we have this reduced lipolysine. Right? Remember, there's a lot of energy there. So we need to pull out those electrons in that lipolysine. The way we do that is to transfer those electrons to FAD to form FADH2. And so in this case, the lipolysine will pass these electrons to FAD, you form FADH2, and then you're back to the oxidized lipolysine here. So lipolysine is restored and ready to pick up another molecule. And we have one issue though. FADH2 is a prosthetic group. So it is bound to this enzyme, so it needs to be restored in this process. And so FADH2 will take those electrons and pass them on to NAD plus to form NADH. Now FADH2 is back to the oxidized form, FAD. The lipolysine is back to its oxidized form, and TPP is all good. And we have formed our acetylated coenzyme. So now, when all is said and done, what do we have? We have a molecule of NADH, and since it is a coenzyme not attached to the enzyme, it can float off to go to the electron transport chain. We have our acetyl-CoA that will go to the citric acid cycle, and we have our carbon dioxide that will leave the cell as waste. And there we have it. Now we are ready. This acetyl-CoA is what we're going to follow through the rest of this process. Oh, if you look at the enzymes, pyruvate dehydrogenase, dihydrolipotransacetylase, dihydrolipoyl dehydrogenase, you can look at the enzyme name and know what's going on. We're dehydrogenating this pyruvate and removing the carbon dioxide from it. Dihydrolipoyl transacetylase, so we're taking a dihydrolipoyl molecule and acetylating it, and then dihydrolipoyl dehydrogenase, we are taking that dihydrolipoyl model and then we are removing the electrons from it. So the enzyme name itself tells you what's going on. So complicated, I go through it independently, but if you're able to follow where your pyruvates are, if you're counting your carbons and following the electrons, you're good to go. We talked a little bit about poisons and irreversible inhibition. Arsenic irreversibly blocks this lipolysine and prevents it from being able to pass on those the acetyl group, therefore blocking your ability to pull energy out and taking you out. Okay, I'll stop here. We're now prepared to go into the citric acid cycle, and so I will end this and then we'll pick up to do citric